to the second question. And some of the team going to swap in and out, as in David's going to swap in for this one. But I don't know whether... No, you're staying in. You've already volunteered for both. And Bart, you volunteered for staying. I think Sandra's staying in. Is anybody else that I asked to or would like to come down? So the topic is whether neuroinflammation is good or bad. Does anybody have a strong opinion about it? So you'd be surprised that actually people have quite strong opinions that microglia are always good or always bad. It seems obvious that they would be both, but it's not necessarily true that they would be both good and bad. And people do have very strong opinions. For example, whether DAM, disease-associated microglia, are good or bad. It's not entirely clear from the evidence as far as I'm aware, but perhaps we'll get some enlightenment from the panel as to whether they think neuroinflammation is good or bad and in what kind of conditions or in what ways. Perhaps we could start with Michael.
I know it. No, so the first question is, what is neuroinflammation? And how we define it, and is it an inflammatory reaction? Is it the release of any inflammatory mediator at any concentration? Couldn't we imagine that low level is enhancing resilience at some stage, and high levels are detrimental, or is it the acute versus the chronicity? I think I'm mostly working on Alzheimer's disease, which turns out to be a almost lifelong problem. It starts somewhere in the third or fourth decade of life. It definitely runs 3 decades prior to the onset of dementia. I don't think there is one type of neuroinflammation in this disease. I think this goes in waves. I think this is way more complicated. The disease evolves and it goes from one brain, kind of spreads from one brain region into another brain region. That's one level of complexity, and the cells reacting to it as well. Now, in our own work, we had when I was in Bonn, we started 10 years ago a longitudinal cohort called DELCODE. So we take CSF when patients are enrolled, and then we follow up every year by a 7.5 Tesla MRI and by neuropsychological analysis. And so we started to look at a panel of inflammatory markers, and this is published data last year in Neuron. And it turns out that there is almost at the beginning of the first biomarker change, classical biomarker changes, there's an so-called inflammatory signal by TAM receptors, soluble AXL, soluble TYRO3 go up, and the soluble levels, so indicating that type of inflammatory reaction of the microglia cells, is to our big surprise associated with a stable cognition over the subsequent years and no brain atrophy. While that part of the population that has a low level of both markers proceeds into atrophy and into cognitive decline. So in a blank answer, not every type of a microglia reaction or a microglia activity is detrimental, for sure not. But I believe from own data and from data from all of us that once this becomes chronic and persists over years and decades, and maybe also kind of fights down resilience factors and others, that this certainly is something which ultimately is detrimental.
David. Yeah, thanks. I think I just want to pick up on something that came up earlier from Andrea Blasser's talk. And she mentioned very briefly antagonistic pleiotropy, which is an evolutionary theory of aging. And I think it works quite well when thinking about inflammation. So our inflammatory response is necessarily severe because it's got to kill pathogens or kill pathogen-infected cells. And the antagonistic pleiotropy is because we've got to survive through reproductive ages. And that's the evolutionary pressure for that. when we get older and then diseases of aging, that evolutionary pressure isn't there, and we still have this severe response to disruptions in tissue homeostasis. So the immune system's reacting as if there's a pathogen. And I think in almost all cases of diseases of aging, whether it's chronic like Alzheimer's disease or Parkinson's disease, there's strong cases for inflammation being very damaging. Even acute conditions such as stroke, we've done a lot of work on, you get an inflammatory response to the tissue injury, it's very damaging. You block inflammation, you have protection. And yet, we know neuroinflammation, for example, sickness behavior, which we touched on earlier, is a protective response for a younger organism. But maybe, as we mentioned, if that sickness response was to interact with a pathology in later life, that could be detrimental. We know that infections predispose people to stroke risk, they make strokes worse. We know MS is worsened by bouts of infection and so on. So I think it's a question of age and context, and I think certainly in the brain and diseases of aging, inflammation is a bad thing.
Yeah. Yeah, I think it's neuroinflammation. Yeah, it's complicated. What Michael says, I think it's what do you classify as neuroinflammation, and I think it matters on the time, the duration, the challenge, the frequency, the extent, and the same challenge at a different moment can have a different response. So I think it's all context, time, frequency, and intensity dependent. And then sometimes it's good, sometimes maybe it doesn't matter, and sometimes it's bad. And for instance, I do some MS work, transcriptomics. If you have a lesion and your myelin is degraded, in order to repair it, you need to eat it. So microglia need to become inflamed or whatever, or activated, to use a very bad term. So they need to eat up the myelin in order to set the stage for OPC recruitment, maturation, remyelination. So you need these cells to be sort of inflamed, you need neuroinflammation to clear up the damage, and then you need a different response to repair the tissue. So I think in some contexts it's good, but if you have a persistent inflammatory activation of these cells, I think that could be potentially bad. Like Alzheimer's is a good example where you have this constant trigger around. So the thing is, as I said, I'm repeating myself, context, time, intensity, dependent.
I can only second all what has been said. For me, the question is also like, is it good or bad? I cannot answer. It is. I totally agree with everything what has been said before. Also like would to think about another factor which I would like to bring is also the sex as a potential factor in inflammation response, because that is often undermined. And I'm often shocked also that I see studies where it is not described which sex a certain data point is. Or it's written even worse, they say we use mixed sex, and then I look at the transcriptome data, and there you can actually figure out in single cell analysis which cells are female or male, and then you see that the data set is totally unbalanced, that the one time point is actually only female and the other time point is only male. And I'm like, okay, I mean, coming back to this question, is this like, what can I learn then from this data set? If I'm like a totally naive one, I'm like suddenly struggling and would make the wrong assumption and say, oh, this is a general phenotype, but actually this is a phenotype that happens only in female, potentially. So I would like also to raise awareness that, especially in the context of inflammation, sex is a critical factor that has to be, no matter what, notated. And ideally, if you show a graph, you show like in circles and triangles what is a male and what is a female, and maybe you even see like a disbalance, which would be an interesting information for the community also. You don't necessarily have to address it, but in it itself, of course, a reviewer will probably pick up and say you should do ovarectomy, which I said that you should not do because we don't know what phenotype we have there. But I think it is important that we open up to this perspective, because otherwise we restrict ourselves and limit our possibility to learn something valuable.
Went to a meeting organised by helmet ketamine, and there was a guy there, and he said, I was very new to the field, and he said, "Oh, microglia, little bags of poison," and I remember thinking, how can that be? I mean, why would you have that in your brain? You know, you have something in your brain which is protective, but then, as we've heard, you know, acutely neuroinflamed, but not really. I mean, neuroinflammation has a kind of nasty connotation if you think about it. People think, oh God, you know, it's really bad. But initially, I don't think, you know, you need that inflammation. You need those cells to be able to respond. But as it becomes repetitive, as it becomes more chronic, then they become dysregulated. And so that is the, you know, what I would call the sort of neuroinflammation, where they no longer can control or be downregulated. It can't be suppressed, basically. They cannot go back to this state where they can kind of respond and downregulate and respond and downregulate. They just simply can't. It's a bit like the sort of MS line that, you know, relapsing, remitting, and then to a point where it simply can't. So I think they're there for a protective reason. And we all know that very, very early stages of AD, there is synaptic loss. What is that? I suppose we still don't really know whether that's due to the fact that the microglia suddenly decide to munch them, or whether there's something cognitively going on wrong with those synapses that microglia start stripping them. We really don't know. So I think it's, you know, they're there for a reason. They're there for protection, but they go awry as you age and as you get into a newer degenerative disease. So that's my view. What do we all think? Yeah.
It's a very interesting debate. Thanks for organising this. I mean, I guess one thought immediately is about language, because you're all kind of using the words inflammation or neuroinflammation and then forgetting that actually the immune system is a multi-cell dynamic process when we concern ourselves maybe just about microglia. So should we just be talking about that one cell type and not using some of these terms interchangeably? Because again, the important thing there is the interaction with other components of the immune system, which were somewhat alluded to in another mention. So in MS, you may well have ingress of T cells that are shaping the microglia phenotype, so for example. And then just from hearing the comments, which I sort of also fully agree with, it just emphasizes the importance of longitudinal data. So these observations coming back from clinical populations and then asking relevant questions like using appropriate model systems to see how these things are dynamic, how they change over time. Is it sort of in the early stages, as Jenny was saying, if you see changes in microglia, is that just a reactive response to some neuronal dysfunction, and then as they try to compensate for that, they proliferate, and then that induces another problem, and then later on there's some other blood-brain barrier breakdown, something else happens, which is all going to be different in sort of space and time. So I just wonder if we can somehow, is there a way we can design experiments to sort of tack on to, Michael, you mentioned this beautiful longitudinal clinical study. Is there some way we can leverage some of these biomaterials to, I don't know, like make better models? I mean, not necessarily iPSC, because we don't want to go right back to pluripotency, but transdifferentiation approaches, something we didn't discuss. I don't know, just some thoughts.
Did anybody want to respond to that?
If I could just comment on looking at the other immune cells and the interaction between innate and adaptive immunity is absolutely important. And I don't think we're considering it's the purely microglial effect. The microglia can set it up, but it's, there's a lot of evidence of other immune cells getting involved and being recruited to lesion sites and so on.
Yeah, I mean, that is an important point, what we mean by neuroinflammation, as Michael says. And as he said, that there's a group of people who think you shouldn't use the term at all unless there's ingress of immune cells such as neutrophils or T cells or monocytes. we shouldn't be using the term. Does anybody have any opinion on that?
I think it's probably the question, because we know from the cross-sectional analysis that there are many different cells. You know, the term of border macrophages has been coined. We have, if you ask David Gate and Tony Viscore, they will say there are a lot of T lymphocytes. Ludwig Eigner's lab has provided evidence for that as well. There are T lymphocytes. They're most likely to shape what, again, all microglia cells or a subset will be doing. We know that there are dendritic cells. We know about perivascular immune cells. So it's way more complex. And again, we just discussed that before. Microglia cells are not one population. They differ. We know this already from cross-sectional analysis. How will that look if you do a longitudinal analysis? So we have an almost endless complexity, but I think we need to start at some edge and then walk our way. But we should be aware that it's exactly right what you said. I think this is not about microglia cells. If we speak about neuroinflammation, it involves way more cells, and maybe also neurons. We know that neurons react to and neurons feed back by, as we discussed, neurotransmitters, for example.
It's quite interesting question, and I completely agree with all the panel member what you said. It is all time dependent, and the state of the the neuroinflammation and the age or the disease stage. And as Jenny said, we came through the evolution, and why would we have these poison bag in the brain for so long if we don't need them? So obviously, in context of neuroinflammation is complicated, but in context of response to the neuron state, microglia or other cells reacting to that response and producing neuroinflammation, considering only that scenario, I think obviously it is beneficial at the start, as we all agreed, or again, it is a disease state dependent, and in later phase it becomes detrimental. But I think ultimately what it comes in the somewhere in that mid time, neuron state changes, and that neuroinflammation become detrimental for neuron, not per se, maybe the cells that are producing neuroinflammation, they are doing at the acute or chronic stage of the disease. It's about the neurons state that time. And I just wonder whether it would be essential to focus on the neurons at which time point neuroinflammation is actually good or bad to them.
I would like to respond. I agree and I disagree. And I disagree by citing Darren Baker's paper published in Nature, October 2018. There is first author is Busjan, and what they showed is if they induce senescence in glial cells, they drive tau pathology in the neuron by solely changing the senescence associated secretory profile of microglia cells and astroglia cells. So we cannot just look to one. So it's a clear-cut paper, it's no doubt. And if you look to Christina's Ising paper a year later from our lab, there's similar data. So we cannot focus on the neuron. The neuron is the sufferer in one term, but it's also one of the evildoers in that game. I just believe that we cannot focus on the neurons alone, because obviously the changes on the glial cell compartment is very important of what happens on the neuron side.
Do you think as the discussion started, that how you define neuroinflammation? Because when you say inflammation, in general, inflammation doesn't carry that negative connotation. But as you say neuroinflammation, people start becoming really biased that it is good or bad. Because even when you go to lab, start doing research, generally the question is introduced to you, neuroinflammation in case of disease, Alzheimer's disease, Parkinson's disease, and generally the aspect is that how it is killing neurons or how it is affecting microglia in a negative way. You will rarely find a paper which says that microglia only in healthy humans, what's role and how it is acting. It is only in health and disease. But inflammation you do found in textbooks, and it's established that it is good for you. But neuroinflammation in our textbook, you will not find that statement that it is also good for you. It is also always in context of disease. So is there any way to that, how we convey it and how to introduce it?
I would argue that in a normal homeostatic resting brain, microglia are sort of within a certain bandwidth of responsiveness, but if they start secreting inflammatory molecules, that is usually in response to some sort of damage, trauma, disease. So I think that's why that phenotype is often described in the context of what you discussed, but it doesn't necessarily mean it's a bad phenotype.
I'm maybe not answering directly, but just a comment. I think when we also think about neuroinflammation or in disease, people that have neurological or neurodegenerative disease, I don't think we can look at it in isolation as just the brain. People often have comorbidities, cardiovascular disease that are very inflammatory. They're old, which is inflammatory. And there's and all this contributes to disease in other tissues. And if we consider if that's a target, if inflammation's a target, there's going to be consequences to targeting particular processes that will ultimately affect other parts of our physiology. And so we might be, using something to target inflammation in the brain specifically, but then we're making ourselves more susceptible to the ravages of some other disease or infection, which may in turn have an impact. So I think it's about maybe not just looking brain only, but looking at the influence of other body systems on the brain as well.
Yeah, I actually would like to bring together this part of the discussion to the first part about the transcriptomics. What we see is that in the times of single-cell sequencing, often quite minor subsets of cells are actually blamed for doing something wrong in the inflammatory context, and not only microglia, also T cells. And it's not very easy to be sure about these findings, and I mean how reproducible they are. And I'm wondering what can we do to validate these basically findings coming from single papers describing very small subsets of cells that are detrimental. And actually exactly the same in the T cell field. That's why I'm also asking here.
Bart. Yeah, I lost the reasoning a bit. Could you?
It's about the single-cell sequencing, which quite often defines very small subsets of cells that have detrimental activities described in one single paper, and then the reproducibility of these findings. That is something that concerns me a bit, and that is something what I now also see in not only for microglia but also for lymphocytes infiltrating the brain. How can we handle that? What is necessary to confirm these findings?
Yes, I find a difficult question, Jörg. Also, I don't know, but maybe indirectly a little bit. So when we try to build this website and add data, publish data by other people for others to access, that you don't need bioinformatic expertise to do this, we had to dismiss a lot of studies because there was like NS1 or super shallow sequencing, a very low number of genes detected. So I think that is part of. It's getting better, but I think the standard there should be, and I think it's good that you have now star methods, or that you really need to add what did you exactly do and what is the quality of your data. QC metrics are crucial, and I think if that standard that's get more standardized, that would help in the reproducibility. Sorry, I think, but. In the early days, I think also the technology was not so advanced. That might be underlying. And then also low. I mean, the first papers, if you look, there's only a handful of cells that were sequenced. Now you have to come up with 500,000. So it's a numbers game. It's a robustness of the platform. How is it done? Yeah.
Okay, I think the last question.
Yeah, I guess I'm thinking something really obvious, but I'll just say it anyway. I guess the genetics is telling us that there are protective variants in genes as well. You know, we've heard nice talks already from PLCG about PLCG2, CD33, SPI1 is also protective, and then there are Christchurch mutations, RELIN. I guess we understand less about those, but they clearly seem to be protective. I guess there's some interesting hints there about what they're reacting to and how they drive disease. You know, for example, they seem to still have amyloid pathology, and it seems to be the tau conversion that's different, and other things like that. So I guess we might gain more by studying some of those protective variants. I guess they're harder to study. You know, tissue's less available as well.
Great. Does anybody want to respond to that?
No,
Yeah.
he's totally right.
We have to do more work. I completely agree. Yes. Yeah.
Great.
Mainly I can add an example which goes into the same direction. I was involved in a study looking at supercentenarian Italians which have an outperforming good cognition. And then I got a call from the study lead saying, Michael, you're totally wrong. These people have higher inflammatory molecules by about twofold, 1.8 to twofold higher increase to most of the pro-inflammatory mediators we do know, including complement prostanoids, IL-1 beta, TNF-alpha, IL-6, and so on. That's the one I remember. And then it turned out I got a call about 8 weeks later that, yes, that's right, but the anti-inflammatory molecules and the anti-inflammatory signaling were up by 10 times. So we often, and I remember when we measured prostaglandin E2 because R&D had an assay which was, and it was probably one of the only and earliest inflammatory markers we could determine in the early 90s, and so we have this little bias towards the pro-inflammatory molecules, and a lot of studies miss looking at the anti-inflammatory side at the same time. And this is exactly what you say. I think genetics teaches us to do so. And so I think we have to consider that when we speak about immune activation, inflammation, this is always a balance about a number of things that drive into the detrimental and drive into the beneficial pathway.
Great, thanks very much. So I think we've learned the answer to the question whether neuroinflammation is good or bad is it's complicated, which is a good thing because it means that we've all got some work to do in this field. So thanks very much to the panel.